Okay, Assalamualaikum warahmatullahi wabarakatuh. Today I'm going to present about the diversity of marine phototroph, evolutionary of marine phototroph, traditional to the current times. The terminologies. Phototroph are organisms that can synthesize ATP using light as the energy source and can perform photosynthesis. And directions of photosynthesis is the reactions that can reduce carbon dioxide into organic compounds such as sugar using sunlight as energy source. Photoautotroph is autotroph. It's making its own food. Organism that can synthesize its own food from inorganic materials using light as a source of energy. And then photoheterotroph means gain food from the other organism that use light for energy, but it cannot use carbon dioxide as its sole carbon dioxide. It's take organic compounds from the environment. Primary photosynthetic organism that can produce organic compounds that from inorganic carbons and that can produce ATP using sunlight energy. Okay, this is basically the flow from traditionals to the current understandings of diversity marine phototroph. In the mid 1970s, the atoms and flagellates are primary producer in marine environments. Okay, I'll show you guys the image. Okay, this is the atoms and this is a flagellate. Okay, a major organism that can be collected by a phytoplankton. This is a phytoplankton. Okay, the receiver, which is we call it a cod end, where the phytoplanktons will be trapped inside it and we can collect all of it. In 1970s, net with mesh size of 100 micrometer to 70 micrometer and 60 micrometer will use only phytoplanktons. Okay, only phytoplanktons with size of more than 60 micrometer will be trapped inside the phytoplankton. So this is a phytoplankton net, the real life. Dinoflagellates and corals at tropical reef play a significant role as a major primary producer in the oligotrophic environment. This is dinoflagellates. Okay, what is oligotrophic environment? Oligotrophic environments is where the environments with a low nutrients inside the lakes and with a high oxygen levels. Okay, and there is a eutrophic zone where it was low with oxygen levels and there is abundance of algae. DNA detection and flow cytometry has shown that the predominant phytoplankton in marine environment is much smaller than those collected by phytoplankton. For example, Coccolithosaurus. Okay, this is called Coccolithosaurus. It is smaller than the atoms and the flagellates. Okay, flow cytometer instruments for rapid detections and characterizations of microbial cells based on their light scatter and fluorescence properties. Okay, basically they're using an antibody combined with the target cells, and then they're using a laser to detect the cell fluorescence. In the late 1970s, fluorescence microscopy with electron microscopy observation revealed that unicellular cyanobacteria, Cyanococcosis, are predominantly distributed in the marine environment. Okay, they found a Cyanococcosis. This is the fluorescence microscopy with electron microscopy. This is a Cyanococcosis. Cyanococcosis play a significant role as a primary producer because it was predominantly distributed in the marine environment. They are very tiny cells with 0.5 to around 2 micrometer in length and 0.5 to 1 in diameter called Picocyanobacteria. It was reported in 1979. Okay, ABB can be classified into aerobic and anaerobic. Aerobic, it's need oxygen, with oxygen, needs oxygen, and the anaerobic without oxygen. Okay, actually ABB, anoxygenic photosynthetic bacteria, it don't produce oxygens, but oxygenic photosynthetic bacteria it will produce oxygen. Okay, but this AAPB and NAPB both have bacterial chlorophylls for their photosynthesis. Actually, aerobic anoxygenic photosynthetic bacteria is derived from non-cephalopus anaerobic in the alpha proteobacteria. Aerobic is a photoheterotroph. It contains about 1 to 24% in a euphotic zone, and the anaerobic is 30%, 30% in some specific heats. It's quite a lot, so it's considered as a primary producer. Next, proteorhodopsin is detected in marine proteobacteria. Retinolopsin complex is a member of rhodopsin. Okay, I'll tell you guys what is the rhodopsin. Okay, proteobacteria is actually gram negative. You can see the differences between the gram negative and positive. It's in the layer of peptidoglycans and the outer membranes. Okay. Wide variations of rhodopsins like genes were detected in seawater by metagenomic analysis. Metagenomic analysis is a study of the genetic material from a mixed community organism. The total amount of proteorhodopsins containing bacteria is 13 to 80% of the total amount of bacteria and archaea in marine environment. Thus, it plays a significant role in light energy transformation. Okay, some examples. Bacterial hydropsins is halobacteria. Halobacteria we can find in the very saline water, such as a Dead Sea. Okay, that's a light-dependent ADP synthesis using proteohydropsins was reported in a Flavobacterium strain. Rhodopsin is consists of protein, which is contains of opsins and retinols. Opsins is a bundle of 7 membranes alpha helix. A aldehydes bind to the lysine residue at the centrals of the opsins. Opsin is functioned to absorb light as it G protein. Absorptions are visible light, but the chromophore retinol itself has an absorption maximum in the UV regions. Rhodopsin is well known as an alpha photoreceptor in invertebrates as a light sensor. In 1980, Picocyanobacteria dominans, developments of HPLC. HPLC, this is the image of HPLC, okay. Technique for HPLC is a technique for pigment analysis and of the cell detection technique using flow cytometry brought new insight in marine picocyanobacteria. So by using HPLC, we can discover more about the picocyanobacteria. Senecaca species contain divinyl chlorophyll A and no phycobilipigment. What is phycobilipigment? Phycobilipigment is actually an accessory pigment commonly found in the cyanobacteria or in the red algae. The function is for absorb light. Chlorochloroscoccus is actually a cyanobacteria, unicellular with 0.4 to 0.6 in diameter and 0.5 to 0.8 in length. As Prochlorococcus, it's perform oxygenic photosynthetic means it produce oxygens using divinyl chlorophyll A or B, which it can efficiently absorb in the blue light dominating in the ocean. Chlorococcus, it's dominate in the tropic and subtropical about 30 degree south and north. Okay, between these, okay, you can see I have highlighted. In several regions, the amount of divinyl chlorophyll A is 3 times higher than the chlorophyll A. And it means that in the regions, this Prococcus is a primary producer because Prococcus contain divinyl chlorophyll, right? We can consider that because divinyl chlorophyll is found in Prococcus. Okay, so we know that Prococcus is a primary producer. In 1996, oxygenic photosynthesis using infrared light, chlorophyll A is indispensable for oxygenic photosynthesis and it plays an essential role in light harvesting, energy transfer, and transpiration in the primary reaction in photosynthesis. Okay, chlorophyll A actually it will be found in all plants, algae, cyanobacteria, which under photosynthesis because chlorophyll A is the primary pigment. Amounts of oxygenate phototroph are estimated based on the amounts of chlorophyll A. Okay, this is AR photosynthetically active radiation is used for the evolutions of light strength for the activity of oxygenate photosynthetic. Cyanobacterium Alcayocloris marina perform oxygenate photosynthesis using chlorophyll D. Okay, we just found chlorophyll D, okay, here in 1996. Okay, chlorophyll D, it's absorbed far light longer than chlorophyll A. Okay, Acariochloris marina we can found in the coastal area as epiphytes on seaweed. It's a leaf on the seaweed. We can see, okay, there is a yellow color is a cyanobacterium of Acariochloris marina. Okay, amounts of chlorophyll D on the seaweed range between about 1 to 13% versus total chlorophyll A means there is an abundance of chlorophyll D means it's a lot of Acariochloris marina. Okay, in the early 2000s, a new finding reported that there are microorganisms other than phytoplankton can transform light energy into chemical energy. This in the past years, we know that phytoplanktons and plants, but then there are other organisms that can transform light energy into chemical energy. In 2000s, what distributions of aerobic and oxygenic photosynthetic bacteria, AAPP, just now we mentioned, contain bacterial chlorophyll A, mainly recognized in anaerobic photosynthetic plants. Proteoherbicin containing bacteria, okay, there's a wide distribution of these. Both bacteria are photoheterotroph without carbon fixation. In early 2000s, eukaryotic phytoplankton species have been revealed, just some of them. Okay, for examples, species in Chlorophyta, Heterocontophyta, and Haptophyta. In 2007, phytoplankton, which did not belong to any phyla of algae, were discovered by metagenomic analysis of a plankton fractions of less than 3 micrometer in size. The picobellids distribute widely in the coast area of Europe and the North Atlantic, and the same number was reached up to 1 to 6% of the total eukaryote phytoplanktons. Okay, Picobellids DNA sequence was close to the Cryptophycines lineage. Okay, you can see this is Cryptophycine. In 2010, another redshift chlorophyll F. Okay, in 2010, we found a chlorophyll F. Just now, we found a chlorophyll D, which is also a redshift chlorophyll. Chlorophyll S was reported from a cyanobacterium isolate from the microbial mat on stromatolites at the coast of Australia. What is stromatolite? Stromatolite is deposition produced by the sediment trapping, binding, or precipitation activities or microbial communities dominated by photosynthetic bacteria from this cyanobacterium. Actually, stromatolite is an early fossils. Okay, there's only 2 well-developed marine stromatolites area in the world. This is what I found. Bahamas and Himalayan Pool in the Shark Bay area of Southern Australia. Okay, this is not a coral. It was a stromatolite. Can show this. Okay, chlorophyll F was absorbed maximum wavelength at around 700 and absorbed far red light up to around 760 in people in the living organism. Marine phytoplankton analysis using the technique of metagenomic and flow cytometry revealed that picohaptophytes, including many undescribable species, were widely distributed. Keratonite 19HF was predominantly detected in the picoplankton fractions less than 3 micrometer at a high latitude area of 40 to 60 degrees south and north. Okay, 19HF is a specific pigment of haptophytes, which contribute to 50 of picohaptoplankton fractions and 5% of the global biomass. It was considered as a primary producer. Reported that a significant amount of microalgae in an unknown phylum also exists in marine environment in 2010. In 2011, it was a strange phototrophic, which monads, okay, also widely distributed in coastal area of Europe and the Atlantic and Pacific Ocean. Monads have DNA that is distinctly different from that of other known algae. Rabomonad is basically name of a professor from University Hawaii, which name is Michael Rabe, who was the first author of the paper which discovered this microscopic algae by following up on an unexpected DNA sequence list in a research paper from the late 1990s. This strange phototrophic monads is detected by metagenomic analysis target plastic DNA. The cyanobacteria-like particles used YNA was studied by metagenomic analysis also by metagenomic analysis of nitrogen-fixing bacteria in marine waters. Okay, using YNA, it had a nitrogenous gene for nitrogen fixation in pelagic water. Okay, so usual cyanobacteria grow by an oxygenic photosynthesis, process photosystem, a respiration system, and genes for air carbon fixation system means it's produce oxygens. Okay, but UCONA, it's like photosystem too. In a photosystem too, it will produce ATP and release oxygen. It's lack of that. For the system 2, it's lack of rubisco, it's lack of the primary enzyme for fixing carbon dioxide and the TCA, 3-carboxylic acid cycle for respiration. Isocyanates is symbiont with a small planktonic unicellular haptophyte algae. Isocyanates is an unusual cyanobacterium lineage that has a lot that have lost many of the typical cyanobacterial metabolic pathway. Looking at the algae, alga in a high latitude water means in a cold water. It's will found urine A. Since this possesses a complete chain set for photosystem 1 and contains chlorophyll, it must use light energy for some activity of the particles. So we still discover about this. For the traditional pseudocurrent times, and this is a method, basically the method and technique of evaluations in understanding of marine phototroph from 1970s until 2011s, whereas from the planktonet, we use planktonets, and then the detection and flow, sedimentary fluorescence microscopy with electron microscopy metagenomic analysis until the big machines HPLC. Okay, the accumulations of knowledge on the diversity of phytoplankton, including unknown, might contribute to a better understanding of primary production in marine environments and the developments of technology. Okay, that's all from me. Thank you. Okay.